Our webinar from ForceNet. If we haven't met before, my name's Greg Kotchuk, and I'm going to be your host and moderator. And it's really a rare situation where you get to kick off a meeting like this and talk about how awesome Twitter is. But this may be like the one time where I looked through Twitter and found something that really set me aback. And it was this article called The Next Generation of Evidence-Based Medicine by our speaker today, Vivek Subbiah. And it really discussed where we might be headed, right? We think we're kind of at the pinnacle of what we know in terms of evidence and translation and such. But he will show you how, or talk to us about how things are maybe lagging behind in the landscape of clinical methodology compared to some of the technologies. So, Vivek, I'll get you just to swap your display if you could, and I'm just going to speak briefly to your credentials. There we go. So Dr. Subbiah is a physician who is board certified in both internal medicine and pediatrics and completed fellowships in both adult and pediatric oncology. And since starting his research career about 15 years ago at the University of Texas MD Anderson Cancer Center, he has served as principal investigator on over 100 phase 1 or phase 2 trials and co-investigator in over 200 trials, and is known for his leadership in getting most of these to several first-in-human and practice-changing studies. He has authored over 400 peer-reviewed publications, appearing in such journals as the New England Journal and JAMA, and since 2023 has recently moved to Nashville, where he is now the chief of early phase drug development at the Sarah Cannon Research Institute, where he oversees 9 drug development units. So welcome to our group today, Dr. Subbiah. We're really looking forward to your talk today.
Thank you so much. It's a real pleasure and honor to see you all virtually. And thank you for the generous introduction. So again, I'm a medical oncologist and a pediatric oncologist. So again, everything is from the lens of an oncologist. So these are my disclosures. In the brief agenda for today, I'll be talking about accelerating drug development, innovation in trial design, and lessons from successful approvals in oncology in rare indications, rare diseases. In the last 6 to 7 years, I've been a part of getting some FDA approvals in biomarker-driven rare cancers. So I'm going to share some of the experience, patient-centric approaches, and also conclude by next generation evidence-based medicine. Again, all from the lens of an oncologist. And again, before moving on to my talk, I think Dr. Greg Kotchuk asked me where I am, what am I doing here at Sarah Cannon. Again, in oncology space, Sarah Cannon is known, but outside oncology, not many of you might not be familiar with this. So this is who we are, right? Sarah Cannon Research Institute. I moved here to Nashville 2 months ago. I'm still learning the program and place. It's focused on advancing cancer therapies for patients, providing clinical expertise, trial access, patient recruitment. Recently, before I moved here, the reason why I moved here, they entered into a joint venture with US Oncology. So again, the slide needs to be updated because right now they are in 325 locations in 29 states as of last week. And there are 1,300 plus research physicians, all oncology. They do end-to-end clinical research. And our research network reaches anywhere from 1 to 4 and 1 to 5 patients across, through our affiliated sites. So again, I'm overseeing currently the early phase drug development units and also seeing how we can partner with these late phase locations on seamlessly transitioning the early phase studies. One of the bottlenecks in oncology has been a phase 1 study is done, and how can we transition those studies to across for phase 2, phase 3 seamlessly, fastly, in a nimble way in real time. So again, that is my challenge here, and that is my opportunity to work across this network. We also have a site in London, and they have been extremely engaging in terms of thought process and scientific development here. Again, this is our personalized medicine vision. We want to minimize barriers for physicians. We want to democratize access to technology. and at the same time provide clinical trial options to the right patient at the right time. So this is made possible by a company that was also acquired by this Sarah Cannon, a company spun out of MIT that receives all the structured and unstructured data, molecular data, so that we have centrally all the data so that we can deliver genomic-driven precision oncology to every patient who walks in the door across this network. Again, as we are restoring the balance of cancer care in the COVID era, again, we are seeing more and more patients in the later stages because many patients did not undergo screening. They did not see their doctors. So again, we are trying to restore the balance for cancer care in the COVID era. We are also delivering evidence-based shallow medicine to our patients. Again, most of the time, when we see a patient in front of us, a generic patient in front of us, we are offering very shallow medicine even in 2023 because we don't have all the data for the patient right in front of us. So next generation evidence-based medicine has brought significant advances in oncology in the field of rare diseases. And we've made advances in genomic technology, in precision medicine, in AI and machine learning algorithms. So in the next set of slides, I'll be sharing next generation evidence-based medicine through a lens of an oncologist. We are tasked to slaying the nine-headed hydra, which is cancer. Because as you all know, the story of Hercules and the nine-headed hydra, if Hercules cuts off one head, two heads pop up. Again, that's what we see in cancer. We give a targeted therapy to a particular genetic alteration. We see two different resistance mechanisms popping up and so on and so forth. So again, we are tasked to slay the nine-headed hydra. Dr. Doug Hanrahan earlier last year published The Hallmarks of Cancer, The New Dimensions, and has given us in the field of oncology what we need to work on in the next 10 years that include unlocking phenotypic plasticity. Most importantly, he mentioned about polymorphic microbiomes. We have more than 3 billion organisms living inside us that impact how we behave, how in cancer or even in physiology, how the human body behaves. Again, there's so much to learn from the microbiomes. Again, I'm seeing more and more clinical trials coming to oncology, impacting these microbiomes and manipulating microbiomes, giving specific antibiotics so that patients with cancer have better outcomes. How did we treat patients with cancer before the next-gen sequencing era? It was a one-size-fits-all customized approach. It was a pure guesswork. It was looking into a crystal ball. So oncologists compare ourselves to astronomers, right? Because we call the moonshot program, right? The nation announced the moonshot program, meaning that we chose to go to the moon. We want to cure cancer. So we compare ourselves to astronomers. So this is how Galileo looked into Jupiter, right? Using his amateur telescope. Even if you go to Amazon Prime and buy a telescope, we bought a telescope for $60, and we went to Sedona to see the Jupiter's moons and the galaxy. And this is how it looks. This is exactly how Galileo's amateur telescope and the Amazon Prime amateur telescope looks. So my kids, I have a fourth grader who is very into astronomy. So again, I got into astronomy too, looking into these Jupiters. But right now, in astronomy has come afar, right? We have the James Webb Telescope, which can peer into 13.5 billion light years. Again, I can't even wrap around my head what 1 light year. It's 13.8 billion light years into the galaxy. So this is the telescope's first public image, showing a cluster of galaxies, which is so heavy that it warps and magnifies light from distant galaxies beyond it. This is a cartwheel galaxy alongside two companion galaxies, which was created as a result of intergalactic collision. We are able to do targeted interventions in space. This is the DART program by NASA, Double Asteroid Redirection Test. So the Dimorphos was a minor planet of the moon of the asteroid Didymos. The DART spacecraft successfully collided with Dimorphos on September 26, 2022, and shortened its orbit. So we are astronomers, NASA folks are able to offer targeted interventions, targeted therapies in space. Oncology has moved on from the light microscope to the molecular microscope, just from Galileo's telescope to the James Webb telescope. We are not quite there yet in oncology like the James Webb, but we are there. We can say we are there in the Hubble space era, right? So from 1600s, we have moved on to molecular microscope. So what is a molecular microscope? The molecular microscope essentially is a next generation sequencing technology. 2.5 decades, 25 years ago, they said we cannot sequence a human genome. Now we've sequenced a human genome. It took $3 billion to sequence the first human genome. Right now, we can get a human genome sequence, at least a somatic cancer genome sequence for $1,500. Colleagues in India and China say that they can do it for $50. One colleague in Shanghai and another colleague in Mumbai have said that they're going to take it down to $1. This is because of Moore's law. The cost per raw megabase of DNA sequence is coming down. So if it's $1, genomics will be a part of diagnosis in every clinic all across the world in the next decade. But unfortunately, in oncology drug development, we have the opposite of Moore's law. What is the opposite of Moore's law? It is the Erum's law. What discovering new medicines gets more and more expensive. That is Erum's law. 30 years ago, it took $1 billion to develop 30 new drugs. Right now, in 2023, it costs $1 billion to develop one drug, if not more. So how can we break the cycle of Erum's law? In oncology, at least, we are able to think about breaking the cycle of Erum's law by the advent of next-gen sequencing, because we can customize the right patient to the right treatment at the right time. Biomarker-driven drug development. Another thing, what we are seeing more and more in oncology is a tissue-agnostic drug development. I'm going to be talking more about tissue-agnostic drug development because this is biomarker-driven drug development. An orphan and rare disease drug development, and also this accelerated approval program that has benefited oncology and many rare diseases. So these are some of the precision medicines that have been approved in the space of oncology. This slide is already obsolete because we have at least 10 different precision medicines being approved in oncology after this publication. So again, delivering precision therapies to the right patient at the right time is already standard of care in many, many diseases in oncology. So I mentioned about tumor agnostic treatment. What is tumor agnostic treatment? Tumor agnostic treatment is a drug treatment that is used to treat any kind of cancer, regardless of where it started in the body. Oncologists are used to developing drugs for breast cancer, kidney cancer, prostate cancer, lung cancer, etc. A tumor agnostic therapy is a therapy that treats any kind of cancer as long as the cancer harbors a specific biomarker targeted by the drug. This is a different way we think about oncology drug development. So the watershed moment for tumor agnostic drug development was May 23rd, 2017. Again, dates are important. May 23rd, 2017. Why? Because this unleashed the tissue agnostic era. This also represented a marriage of genomics because immunotherapy, pembrolizumab, a drug that targets PD-1, which is an immunotherapy, was approved based on a genomic biomarker for microsite satellite instability high cancers. Again, why this was important? Because this was the first ever in the history of FDA approvals, a drug, an immunotherapy was approved based on a biomarker, not based on a location, based on an organ in the body like lung cancer, kidney cancer. So as long as breast cancer, lung cancer, kidney cancer, prostate cancer, any cancer harbors this genomic signature of microsite satellite instability high, this drug is approved. Why this was important? Because this was a tissue agnostic approval. And it says that FDA can approve a drug based only on a biomarker, one. Number two is approval was not based on a randomized clinical trial. Again, the field of oncology has been stagnant because of randomized clinical trials. It takes 7 to 8 years to read out randomized clinical trials. By the time the randomized clinical trials are read out, the data becomes obsolete. So approval was based on a single arm study and also real world data and retrospective data. That was a watershed moment. The second was the story of picking needles in haystacks. NTRK fusions are seen in 1% across multiple cancers, colon cancer, lung cancer, kidney cancer, both in adult and pediatrics. So two drugs, not one, but two drugs, larotrectinib and entrectinib, were approved in 2018 and 2019. Again, age agnostic, tissue agnostic, as long as the cancer harbors the NTRK fusion. So any cancer, patient walking in the door, 1% across all cancers, right? Again, 1% across all cancers is still a considerable 5,000 to 10,000 patients a year. Doesn't matter what cancer. These drugs are approved. But again, these are like picking needles in haystacks. Again, the marriage story continues. The immunotherapy pembrolizumab was once again approved for another genomic signature called tumor mutation burden high cancers, like tumor mutation burden greater than 10 cancers. Another drug called dostarlimab was approved for adult dMMR solid tumors. The story continues. Belzutifan, right, the HIF-1 alpha drug that Dr. Bill Kaelin received the Nobel Prize, was approved again in von Hippel-Lindau disease. Again, this is germline alteration, and patients get renal cell carcinoma, central nervous system hemangioblastomas, etc. So belzutifan was approved again in a tissue agnostic manner for germline mutations and patients who develop these cancers because of the germline mutation. The last two drugs that received tissue agnostic approval, and I've been intricately involved in the development of these two drugs, and I'll be talking to you more about dabrafenib and trametinib for adult and pediatric patients with BRAF V600 mutant solid tumors. This was approved June 23rd, 2022. So again, you see a theme here. So the field of oncology is moving on from a one-size-fits-all chemotherapy to a personalized medicine era where the right treatment is customized to the right patient at the right time, based on biomarkers, based on next-gen sequencing technology. Again, today, as of 2023 November, post-Thanksgiving, we have 6 medicines approved for 7 different indications. Again, we are still scratching this tip of the surface of the tip of the iceberg here. And in the future, I see more and more drugs being approved in the tumor agnostic space because we don't need to do trials in each and every disease harboring that biomarker. As long as we have the drug active in a biomarker, the drug can be approved across multiple indications. The precision oncology paradigm and rare cancer and phase 1 drug development. Again, these have been enabled by evolution in oncology. Again, from the randomized study to what we call as umbrella studies and basket studies, and platform studies and March studies. So let's talk about basket studies. What are basket studies? Basket studies are histology independent studies wherein does not matter what type of disease the patient has, as long as the cancer or the disease harbors the biomarker or expresses the biomarker, they get the same drug. Again, disease A has, we do a sequencing, we get a mutation, say A. Disease B, there is a patient with mutation A. Disease C has a mutation A. They get the same drug. Umbrella study is different because it's all the same histology. It can be lung cancer. We sequence a patient and they get into different arms. One disease, multiple arms. That's the umbrella study. Platform study is different because these are seamless, immortal studies where arms can be added or removed based on the science. Again, interestingly, during the COVID pandemic, it was a platform study, the recovery study, that got us the quickest answers in the world. Recovery was a platform study that was already ongoing in UK, and they added 2 arms to the study. One is a decadron, and one is hydroxychloroquine. So again, to get a protocol up and running, we all know that it takes 6 to 8 months at least to design a study, get a protocol up and running. To get an answer within 2 months in the peak of the pandemic, that was possible because of a platform study, because they immediately added an arm with decadron and an hydroxychloroquine and showed that decadron was effective in patients with acute COVID-19 illness, and hydroxychloroquine was not effective in patients with acute COVID-19 illness. Future, I see more and more of the MART studies, what master observational trials, right? So what are MART studies? These are real world evidence and real world data studies. And these real world evidence and real world data studies are used to build synthetic control arms for natural history of disease in rare cancers. Let's talk about BRAF. And the next set of slides, I'm going to be talking about my experience, my personal experience in the last decade of getting drugs approved in rare biomarker-driven diseases. So one thing I need to mention that, again, I'm in an institution where we do only clinical trials, but fewer and far between. Only 2 to 3% of patients with cancer are enrolled on clinical trials. If we need to move the cancer needle, we need to go up on the clinical trial enrollment. Unfortunately, the real world patients do not have access to drugs without an FDA approval. So the next set of slides, let me show you how we were able to get FDA approvals based on rare biomarker-driven diseases. So BRAF is a gene that is, BRAF V600 is a gene that is expressed in multiple cancers. Again, in hairy cell leukemia, it is 100%. Non-small cell lung cancer, 2 to 5%. Again, rare diseases like Langerhans cell histiocytosis. It's seen in colorectal cancer, hepatobiliary cancer, in pediatric cancers, in tough to treat glioblastoma in adults, in gliomas in pediatrics, Wilms tumor, many tumor types. So we designed early in the decade a histology independent basket study called the vemurafenib basket study. Again, simple idea, right? Any patient with any cancer who does not have any treatment options and who exhausted all treatment options walks in the door, we sequence, and if the patient harbors a BRAF V600, gets this drug called vemurafenib twice a day, oral drug vemurafenib twice a day. We enroll them in different baskets. That's why we call it a basket study: non-small cell lung cancer, advanced disease, rare cancers, among others. Interestingly, the more patients we tested, the more we saw BRAF in lung cancer. The response rate was 42%. There was this rare disease called advanced disease or Langerhans cell histiocytosis. 40 to 50% of patients harbor the BRAF V600 alteration. The response rate was 43%. So based on one of these arms of this basket study, we were able to secure a US FDA approval. Again, beyond these, we saw responses across many rare cancers like xanthoastrocytoma, anaplastic thyroid cancer, cholangiocarcinoma, salivary duct cancer, among others. So this shows the data set of that single of Langerhans cell histiocytosis or advanced disease. And this approval was based on a sample size of less than 25 patients. Single arm study, less than 25 patients. So again, we were armed with this data set. This is a waterfall plot in oncology. So what are these waterfall plots? These show the percentage of shrinkage of the maximum tumor diameter. So if it is less than 30%, we call it a response. If it is less than 30%, it's called stable disease. And if it is above the line, that means the patient's tumor is not responding to treatment, and that we call it progressive disease. Right? So again, you can see the color coding. BRAF is a pan-cancer target. We showed that vemurafenib is efficacious across many non-melanoma cancers. Does not matter what cancer, pancreatic cancer, colon cancer, histiocytosis. As long as they harbor the BRAF alteration, the drug is active. Although we were buoyed by the thrill of success, immediately what we found that patients started developing resistance mechanisms in 4 to 6 months. We found out the resistance mechanism was driven by this pathway called activation of the MAP kinase pathway. So we added a second drug called MEK inhibitor to overcome that resistance mechanism. And that became standard of care. So we designed the next generation of the basket study for BRAF called the ROAR study, the Rare Oncology Agnostic Research. So why did we call it the ROAR study? Because I'm a major fan of Lion King, true story. July 19th, 2019. I'm good with, I don't know, I'm somewhat oddball, I remember dates very well. July 19, 2019, I yanked my kids from preschool to take them to the photorealistic version of Lion King. All right, so Lion King broad study, what simple study design. Any patient with any rare cancer that harbors the BRAF V600 alteration will get two drugs this time, dabrafenib and trametinib. So the first basket was ATC. ATC is anaplastic thyroid cancer. Anaplastic thyroid cancer is one of the most lethal cancers known to us. If a patient gets diagnosed with anaplastic thyroid cancer, they live for less than 6 months. If we see an anaplastic patient who is living beyond 6 months, we call our pathologist and ask them to go back to the pathology and see if they have any papillary component or differentiated thyroid component there. That's how the disease was. That was a bad disease. And the next cohort was cholangiocarcinoma. We also included low-grade and high-grade glioma, tough-to-treat glioblastoma, brain tumors as well. This was the first basket, the anaplastic thyroid cancer. You can see the waterfall plots. Every patient we saw a response. We saw a shrinkage of tumors. And the yellow patient was a patient of mine. This patient had BRAF V600 positive cancer and anaplastic thyroid cancer. He was sent to hospice. He comes to our drug development clinic, and we start the trial. A month later, 2 months later, 3 months later, we get the CT scans. It showed shrinkage. He feels better. His tracheostomy tube, because he couldn't swallow, was there. He took it off. 6 months later, he comes to my office. He's like, "Hey, I got a nice funeral symmetry plot, and they are calling me because they are like, I got to pay something to keep that plot. It's not, I need to give it up." I said, "This drug is working. I think you should give it up." 5 years later, he gave it up after a year. He didn't tell me that. He didn't give it up the same day, but he gave it up after a year because he didn't want to pay the fees for the plot. But 5 years later, he's still around. Again, these are the stories that keep early phase clinical trials doctor up and about in the morning, coming to work. Again, promising responses, beautiful shrinkages. So we were able to secure an FDA approval based on that sample size of less than 25 patients. Again, this was the first ever FDA approved treatment for this aggressive form of thyroid cancer. Again, we were armed with success in precision oncology basket studies in rare cancer. Again, the evidence is probably thin here. There's no randomized study, but we know that these are bad diseases. Patients do not live for more than 6 months with this disease. So again, the first 16 patients we published, and by the time we did all the paperwork, we went to FDA, we had 23 patients on board. We were able to secure an approval. Recently, we published the definitive benefit of this combination, and it was easy to get an approval in the US with the accelerated approval program and a final approval. It's challenging in many parts of the world because most of the world, they require randomized studies. Again, we had to convince many many regulatory authorities to use this data set without a randomized study, using natural history of disease for this deadly disease. Right? We were successful. I think we were successful at least in 16 different geographies to get an approval for this rare disease. The next cohort read out, cholangiocarcinoma. We published it in Lancet Oncology, and we included that in the NCCN guidelines. Again, the clinical benefit was response rate was over 51% of patients. Again, glioblastoma, 33% responses. Low-grade glioma, 69% responses. That was the next cohort. So you can see a theme here. At the same time, the NIH NCI match study published the data with dabrafenib and trametinib that showed, multiple tumor types, including aminoblastoma of the mandible, neuroendocrine cancers, showing response to this drug. So we did a pooled analysis of the ROAR basket study, the NCI match, and the pediatric and another pediatric trial with BRAF, And the entire pool data contributed to the FDA approval in a tissue agnostic manner. Again, as you can see here, that dabrafenib and trametinib is now approved in a tissue agnostic manner across tumor types. And this shows the response rates in many, many, many tumor types. And it works in many cancers with the exception of colorectal cancer. And in colorectal cancer, we found that there is an operating resistance mechanism with EGFR. There was a Nature paper published at the time that EGFR was an operating resistance mechanism. So when we add an EGFR drug in colorectal cancer, BRAF is again targetable in colorectal cancer. So again, today in the real world, in the US, any patient who harbors a BRAF V600 alteration is able to secure this drug because of this data set, right? So the date of approval in melanoma was May 2013, right? The date of approval of the same drug was in June 2022, June 22, 2022. It took almost a decade to get a drug that was already approved in one indication to get an approval in a tissue agnostic space. Again, how do I remember the date? Because I was hiking in Banff. I didn't take vacation for like 2.5 years during the pandemic. And I had so much vacation time, I had to sometimes use it or lose it. So like we decided to go to Banff. And this is my wife, who's also an oncologist. Of course, she's the boss of the entire world, at least in my house. And we went to Banff, and I get a text message. We were hiking there up the mountains, and I get a text message once I come down that, hey, we got an approval, and can you be giving interviews to the media? Again, on vacation, I didn't have any decent clothing. I had to go to the gift shop of the hotel where we were staying. Luckily, they had a blazer. The blazer itself was $300, and there was no sale there. So I had to get that to be appearing respectable to the media interview. Of course, I didn't get pants, which was also another $300. I was okay with up top being dressed in a professional manner. So, but you see that like 10 years, it takes a decade to get a drug that's already approved in one indication. It should not be that. Think about those patients that were waiting to get this drug. Think about that patient who did not have access to clinical trial in New Mexico, who had a glioblastoma, a deadly cancer, who could have been benefited from the drug. Think about that patient in North Dakota with salivary gland cancer, harboring a BRAF V600 alteration, right? Who couldn't access a clinical trial, not getting this drug. Moving on, the latest kid on the block is RET alterations in oncology. And I'll be mindful of the time as well. I think we do have time. So I'll talk about RET alterations in oncology, RET alterations in non-small cell lung cancer. And what is RET? RET is a receptor tyrosine kinase required for normal development. Yes, it was in Canadian dollars. That, at least it was not. Yeah, agreed. It was in Canadian dollars. Great, thank you so much. So again, RET is a receptor tyrosine kinase required for normal development. Alterations in RET function and structure and function is required for normal development. And RET is activated by two major mechanisms in oncology. One, we call it RET mutations and RET fusions. So RET was one of the first oncogenic kinases to be cloned in oncology, 1985. In 2012, this was discovered as a driver event in non-small cell lung cancer. In 2017, we had two drugs entering first-in-human clinical trials, not one, but two drugs. And in 2020, 3 years, we were able to secure an FDA approval, right from first-in-human to FDA approval. I'm going to share that data with you. So RET fusions, as I said, are seen, again, across the board, 1% across all cancers. RET mutations are events, driver events in sporadic medullary thyroid cancer and a hereditary form of medullary thyroid cancer. Multiple endocrine neoplasia syndrome patients who harbor the germline alterations universally harbor the RET mutations. Let me talk about one case of the first ever human being in the universe to get this drug RET inhibitor. This was a 49-year-old man with advanced medullary thyroid cancer. Again, he had progressive disease over 7 years with multiple tyrosine kinase drugs. When he presented to my clinic, his performance status was declining. He had 30 bowel movements a day because thyroid cancer has this paraneoplastic syndrome that secretes this tumor marker called calcitonin. They get a lot of diarrhea. And he was looking cachectic, mainly because of this diarrhea. And at that time, we did a sequencing which showed not only the original mutation, the M918T, but he developed a gatekeeper mutation. So again, this was a resistance mutation that we did not have drugs for in the clinic. So at that time, we had this drug called LOXO-292, now known as selpercatinib. We showed high kinome selectivity for RET in preclinical models, and then showed that it showed blood-brain barrier penetration as well. We showed activity in xenograft models. Again, we treated this patient because we didn't know the dose. The clinical trial was being designed. We did an N of 1 clinical trial with single patient IND. We started, you see the last line here, 20, 60, 80, 120. These are the doses because we did not know the dose of this drug. We started at a low dose. We extrapolated the mouse to human dose. We started at the low dose 20. We went to 60, we went to 80 because we didn't know the dose. We were dispensing pills every day in clinic. So within the first week, there was resolution of diarrhea and pain in the first week. There was reduction in the tumor by 54%. And again, you see the tumor markers here. Again, the graph shows the tumor markers. I don't know if you can see my mouse moving, but you can see the tumor markers beautifully going down, the CA and the calcitonin, and the circulating cell-free DNA also coming down for this patient. But if you follow the tumor markers, there is an asterisk here. There are 2 asterisks showing a bump, right? So that was because it was a major event in Houston. It was Hurricane Harvey. 5 days, the whole city was flooded, and I couldn't go to clinic. Unfortunately, the patient could not come to clinic. Unfortunately, the pharmacist could not come to clinic to dispense the pills because we were dispensing pills every day. So I was worried that we might lose the patient, but 5 days later, we opened clinic on Sunday. When the floods receded, we opened clinic, we dispensed the pills, boom, it started coming down. And you can see, responses, right? Again, this patient who was wheelchair bound, going to hospice, almost dying, we resurrected him. And within 2 weeks, he said he wanted to go back to work. And 3 weeks later, he went back to work. 4 months later, he wanted to go on a ski trip to Europe. So we had to get extra pills, make new pills and extra pills so that he can enjoy a 2-week ski trip in Europe. Again, how did I feel treating the first ever RET positive patient on a drug? I felt on first looking into Chapman's Homer, I felt like some watcher of the skies when a new planet swims into his ken, like Stout Cortez with eagle eyes. He stared at the Pacific, and all his men looked at each other with wild surmise, silent upon a peak in Darien. We did the phase 1 study. After that, the Libretto 001 study. Again, the dose that we reached in that one patient was 160 milligram twice daily. The dose, the FDA approved dose, is also 160 milligram twice daily. The phase 1 dose was also 160 milligram twice daily. This shows the waterfall plot, right? The primary analysis set showing that waterfall plot activity of this drug in lung cancer, RET fusion positive lung cancer. Again, regardless of prior therapy, chemo, immune checkpoint, multikinase, this drug was active. In fact, this was in the last line who have exhausted all treatment options. So we started seeing patients, newly diagnosed patients with lung cancer. Again, the response rate went up when we started treating these newly diagnosed patients, 85%. Based on this data set, this drug received the US FDA approval with a single arm study, right? With a single arm study in the peak of the pandemic. The approval was May 2020. This was published August 2020 because although we had the paper ready, we submitted at the same, just before the FDA approval. It took some time in NEJM because they had all the COVID papers. They were so busy with the COVID publications. So this was deprioritized. So we were even thinking about changing the name of this title to in non-COVID-19 RET fusion positive non-small cell lung cancer patients to get some traction from the New England Journal of Medicine. But having said that, The following year, this made it to the notable articles of 2020, despite all the COVID papers. Right now, this drug is approved in US, multiple geographies, age agnostic, tissue agnostic. Most recently, September 21st, 2022, we showed that this drug is also tumor agnostic. Again, it works in pancreatic cancer, lung cancer, kidney cancer, prostate, any cancer that harbors this red fusion. Again, this is one of the first patients with pancreatic cancer that enrolled on the study. Pancreatic cancer, as you all know, this is a really, really tough disease. And this patient harbored the RET alteration and had a phenomenal response. You see the waterfall plot showing 100%, the blue one, that's that patient. Again, but behind every waterfall plot, behind every scan, there is a human. This is Ms. Cuban, one of a pancreatic cancer patient. Again, she has written a nice book, Needle in a Haystack, because 1% of pancreatic cancer. Again, she exhausted all lines of treatment. She was wheelchair bound, almost hospice bound at the time when she showed up, but we found she hit the genetic lottery, and we had a drug, and we were able to get her on this trial in the right time. Again, when she showed up, she said that she wanted to start the clinical trial in 2 to 3 weeks because she was going to get married, and she thought that the clinical trial would make her sick and she might not even make it. So I said we got to start the trial today or tomorrow, and I'm hopeful that you're going to be okay by your wedding, right? So again, she started the next day. 2 weeks later, she got up from wheelchair. She danced her wedding. She had a wig as well because of the chemo. She lost all the hair. She had a wig. She threw her wig. She had short hair. Again, these are the stories that keep an experimental therapy investigator up and awake in the morning for us, right? So again, we worked on a second drug called pralsetinib. Again, same story: lung cancer, thyroid cancer, 73% response rates. And we also showed tissue agnostic activity. Recently, we published it in Nature Medicine. It was easy to publish in Nature Medicine, tough to get into the cover because we put in first human beings there. Nature Medicine wanted to have nondescript patients, although we then generated AI-generated human figures. They said there will be doppelgangers there, so they made us just have these nondescript human figures there. Again, red superheroes have arrived, right? So this drug, you think about that, right? First in human was 2017, and we got a FDA approval in 2020, right? Because we were able to. This is a rare disease, biomarker-driven disease, and we were able to, fast track the trial process right from first in human studies, an amended expansion arm of the first in human study to a basket design to get an approval in 3 years. But in my mind, that 3 years was very, very long. I think in the COVID pandemic, we could show that we could execute COVID vaccine studies within 9 months to a year. I think we should be doing that in oncology and every other rare disease. But unfortunately, that same patient who had a dramatic response to, started developing resistance. But as I said, if cancers follow the Darwin sketchbook, again, we found resistance mechanisms. And we went back to the lab and we found out that selpercatinib and pralsetinib used an unprecedented binding mode docking to the red kinase, and we found gatekeeper and new solvent front alterations. I collaborated with a colleague to secure an R01 for this, identifying this resistance mechanism. And unfortunately, beyond the on-target, we find off-target resistance mechanism. We found an NTRK3 fusion as an acquired resistance mechanism to RET inhibitor in cancer. So again, we see a lot of off-target and on-target resistance mechanisms. Right now, we are working on combination studies and second-generation drugs to go after these alterations. So again, what is the evidence, right? So we are working on a lot of NF1 studies in cancer drug development. So what are NF1 studies? Trial designs where a single patient is the sole unit of analysis. They are a departure from the traditional drug-centric model to a patient-centric model, right? So again, we use standardized PK-guided intra-patient dose escalation, as I showed you in that one patient. They can fast-track cancer drug development, especially in rare tumor types. We can cut short several phases of drug development. Usually, the 3+3 design in oncology takes almost a year to two to get to a dose. But I think with the N-of-1 studies, we can quickly get to a dose so that many patients can be treated at a therapeutic dose level. I just spoke about genomics. This is a story of 6 blind scientists and the elephant. You can see here the 6 blind scientists, gender balanced here, again, gender balanced, each touching a part of the elephant and claiming it is that. So again, I just spoke about genomics, but cancer is beyond that. It is metabolomics, transcriptomics, immunogenomics, microbiomics, proteomics, and beyond. I think for every patient walking in the door, we need to have a comprehensive molecular portrait. The current paradigm, cancer at the molecular level is a malignant snowflake. And the current paradigm is one-size-fits-all approach for all these malignant snowflakes. The future paradigm should be customized therapies for these malignant snowflakes. One of the silver linings of the COVID pandemic, it has given at least thought in the oncology community to moving from clinical trial-centric to patient-centric clinical trials. We are designing direct-to-patient clinical trials. Patients do not need to travel to niche boutique academic centers, especially if they have rare diseases. And we are designing several direct-to-patient clinical trials. Social media has also come up to empower patients for these rare biomarker-driven studies. Ross Wonders and Red Renegades, all these patient groups are there. So if we have a clinical trial, patients are there to connect to each other. All the precision medicine I spoke about is just across the 2 sides of the Atlantic. Australia, Japan, and China were recently added to the map. Most of the world, it's not benefited from this precision medicine advances. Latin America, Africa, many, many, many countries do not benefit from these precision oncology advances. We need to think globally at the same time, act locally. I mentioned about accelerated approvals. So we work with our FDA colleagues to show that accelerated approvals really hit the target in precision oncology. We don't need to be waiting for randomized study to get the data out. FDA is considering approvals so that patients can get earlier access when the randomized studies are ongoing, because usually it takes 5 to 7 years to execute a randomized study. So again, many of these precision drugs are being approved in an early accelerated approval manner so that patients in the real world can get access. At the same time, the randomized studies can be read out. If the randomized study is positive, then the agent can get a full approval. The randomized study is negative, the agent will not receive a full approval. The approval has to be rescinded. Again, I think patients, especially cancer patients and patients with rare diseases, are okay with certain degree of uncertainty with these single-arm studies, at least so that they can get access to these novel medicines. The current timeline for drug development in oncology is 10 to 15 years, which is unacceptable, unethical, in my opinion. Again, personal opinion, again. We need to cut short the phases of drug development. I think AI, machine learning, is helping us design drugs. AlphaFold 2 is helping dreaming proteins that we couldn't even think of, antibodies that we couldn't even think of in oncology. I think the next decade will be interesting how it's going to help with our clinical trial infrastructure and clinical trial conduct. And I think we still use protocol binders, fax machines to conduct clinical trials. So I'm hopeful that AI can help us to assist in cutting short the timelines for drug development so that we can deliver deep medicine to the generic patient sitting right in front of us. We need all the data. We don't have all the data that we have. So what are the key takeaways? We need a collaboration with academia, industry, community, patient advocacy partnerships. The next generation of evidence-based medicine has arrived, at least in oncology. There's been a revolution in panomics. AI-driven drug discovery efforts are yielding best-in-class molecules. Social media and patient-driven molecular testing is changing the landscape of several rare biomarker-driven diseases. These 1% cancers are becoming more and more common. Because, why do I say they are becoming more and more common? Because, if it is 5,000 patients this year, the drug is working. There are 10,000 patients the next year, 15,000 patients following year, 20,000 patients the following year. This is how we are able to convince these companies to develop drugs for rare diseases. Like if you have an effective drug that works, you can convert a rare disease into a common disease within 5 years. Expansion cohorts are leading to FDA approval. So I asked ChatGPT, what is next generation evidence-based medicine in rare diseases? So I collaborated with ChatGPT to write a poem, at least the 8-liner of a sonnet. It's not finished. As with evidence-based medicine, it's not finished. Of course, we know that it has to be 14 lines. I'm working on the next 6 lines. In rare diseases realm, a beacon gleams, next generation EBM, a wondrous sight it seems. With genomic threads, it weaves a tale, unraveling mysteries, never to fail. Precision's mark, it hits with aim so true. Collaborations call a global breakthrough. Patients' voices heard, their wisdom rings, next generation evidence-based medicine, where healing speaks. Thank you so much for the opportunity for me to talk. Thank all the patients, their caregivers for participating in all the studies, all the investigators who participate in the studies. And that's my email. I'm available on Twitter and LinkedIn as well. That's my new institutional email. Thank you so much. Happy to answer any questions here.
Vivek, thanks so much for that fantastic talk. It really opened up, I think, a lot of new lines of thinking for many of us. Some, I'm sure, really exciting questions to come here. And I know that Chad asked the first one that I think probably is on the brains of all of us here, is that when you compare these techniques, whether they be methodologies or statistical techniques and things in rare diseases, what do we have to learn for our group in musculoskeletal diseases, which maybe are more chronic and more common in nature? Do you see these as being very, very specialized things that do not generalize to other topic areas, or are there some lessons to be learned here?
Oh, absolutely. I think, there are some, again, cancer, of course, is a free fall from the sky, right? So we are giving parachutes for these patients. Again, with these musculoskeletal disorders like Duchenne's dystrophy, among others, one thing that you can take away from this is how can you use natural history of disease and using patients as their own control, right? When designing clinical studies, and how do we design better endpoints in clinical studies? Again, we all want for a home run many times, right? Again, what we see, I showed you mainly the home runs, right? I didn't show the 9. Again, I've run more than 100 trials. 5 of them have been successful. I didn't show the 95% of this, 95% of them that have not been successful. But every study, positive or negative, we need to learn from that. Right. So again, one thing is many of the studies that all of us do are negative. We need to learn from all the negative studies and positive studies to design the next phase of the clinical studies. And doing the same thing over and over again, at least in oncology field, and expecting different results has not yielded much success. So again, we are asking the next generation to think differently, to think how we can design studies in a different manner. Again, one thing that what I've seen is we are all working in silos. I think many fields can come together and learn from each other. Again, we can learn all from musculoskeletal diseases, because we see some chronic cancers. We don't know how to develop drugs for those cancers. Because as you can see here, we are able to successfully develop drugs for cancers that are death sentences, right? Like six months survival. But we have cancers in whom patients are diagnosed, they won't have a normal survival, but they live for at least 10 years. We are not able to do studies for those patients because to do an overall survival endpoint there is going to be impossible. right? So we can learn from each other on developing endpoints. Developing musculoskeletal disorders, we are in oncology, what we are right now doing is with the wearable technology, we are coming up with a lot of digital endpoints. So how, because some of these drugs for chronic conditions, chronic oncology conditions, we don't see tumor shrinkages, but they live long. We want to quantify that, right? So they live long, but we don't know how to improve, assess the quality of life. We've administered questionnaires to patients. That's okay. That's patient-reported outcomes. What we've also are doing now is giving them wearable technology and seeing how they behave, how they do, how their activity changes, the delta of activity, and also collecting endpoints. I think we'll be publishing something soon in the next 3 to 6 months on that topic.
Yeah, actually, if I could, I'd like to ask you about that because I noticed, with your waterfall diagrams and such, there's that 30% cutoff that you always had displayed. And I think that's probably an interesting story of how you arrived at that number, the same way we might for certain cutoffs on improvements or not. But you mentioned in case studies how there were particular responses about, say, diarrhea or whatever case may be, but it didn't really see patient-reported outcomes in this. And I was really curious to understand what place it has. So is this a new frontier for you then, bringing in more wearable and patient-reported outcomes into the decision-making process of when a drug is successful?
Yes. The main thing in oncology is what we've done so far as a field is we've used mainly overall survival in randomized studies, objective response rates as endpoints. But many of the, with successful drugs, patients live longer. We want to see how the patients, how the quality of life changes. So we have a quality of life PRO data, validated PRO, patient-reported outcomes data across all these studies. And I didn't show them because of the interest of time, but we do have definite, we publish them, in fact, separately for both lung cancer and thyroid cancer on the quality of life and how it changes with these drugs. But again, these are additional exploratory endpoints. They were not primary endpoints for the clinical trials.
We've got one question here popping up in the last minute or so from Dr. Reed. Do you think that there is sufficient diversity among inflammatory biomarkers to develop precision treatment for chronic inflammation, say in the intervertebral disc versus muscular inflammation? Or are these pathways too generic? Sorry, maybe Bill, you could comment on it, but I think you get the genesis.
I was just trying to look at inflammation, which is common in many diseases, like many cancers as well as musculoskeletal diseases. But could it be possible to develop precision medicine or biomarkers for, let's say, the intervertebral disc versus inflammation in a muscle with there that has, they have different structure, molecular structures and things. And I don't know if you had an inflamed disc, could you develop a treatment for that just for disc tissue versus general inflammation body-wide, tissue-wide?
Oh, absolutely. I think, we need to understand the basic biology, right? If the disc inflammation is driven by one particular pathway, so let's call it the mTOR pathway, right? We can come up with drugs to go after that pathway alone, right? Specific targeting towards that. Whereas if the muscular inflammation is driven by Wnt pathway, right? We can develop drugs for the Wnt signaling pathway, right? So again, looking into the other way, right? If we see inflammation in the vertebral disc or other synovial areas are driven by a particular biomarker, right? It's essentially an agnostic research, right? Biomarker-based research doesn't matter where the inflammation is, as long as it's driven by that particular biomarker, right? And again, any trial that we run, inflammation or cancer, right? We don't have 100% outcomes. Maybe, right? We need to identify biomarkers. So we don't know, right? 10 years in oncology, we did not know what we were treating. We were just shooting chemotherapy in all patients. We're still scratching the surface in oncology, right? So although we have these drugs, right? 90%, although we want to go to the moon, right? We are thinking about moonshot program. We are not testing these biomarkers in patients, right? In the real world. So we need a ground shot here to start helping the real world patients right in front of us.
Thank you.
That's maybe a great way to end it up. I see we're a little over time, so I would just like to thank you once again, Dr. Subbiah, for your insights and the future thinking that you've given us, and maybe just invite you and the ForceNet people to just stay on the line here for a moment afterwards. There's some more ForceNet things coming up in the new year, and you'll be hearing from us about those in the coming days and weeks. But with all that, I'd like to say thank you for attending and appreciate you being here, and have a great day. I'm just going to remove some of the other people here, and then we can just debrief for a second.